Good morning, everybody. I'm Trent Northern. I'm the metabolomic science lead at the Joint Genome Institute, and it is my great pleasure to introduce Gillian Turgeon today. I will give you just a real brief introduction and then leave it to her. So Gillian is the professor and chair of the School of Integrated Plant Science, Plant Pathology, and the Plant Microbe section at Cornell University. She did both her bachelor's and her master's degree at Carleton University in Canada, and then got her PhD from the University of Dayton, but did her research at the Kettering Institute. She's a fellow of the American Academy of Microbiology, and she is well recognized for her leadership in using genomics to study specialized metabolites, especially in fungi. And Gillian, it's just such a great pleasure to have you here today. And I'll turn the stage over to you. Oh, and I want to tell everybody, if you have questions, please type them in the Q&A. This way, if we don't get to all of the questions, Gillian can answer them at the end of the talk. So thank you.
Thank you very much for your introduction, Trent. And I want to say at the beginning, thank you to Igor Grigoriev and colleagues for being our steady companions in our endeavors. My lab works on mechanisms by which microbes, in this case fungi, infect plants. We think a lot about how pathogens differ one from the other, how pathogens differ from saprobes, and we think a lot about why a particular pathogen is pathogenic on a certain host plant. In other words, we think about host specificity of the pathogen. And because I'm likely to not get to the end of my talk, I want to acknowledge the many students and postdocs and visiting scientists that have worked on the projects that I'm going to talk about today, starting with acknowledging Olin Yoder, who is shown at the end of this line, who's started the project before I came to Cornell. And more recently, Scott Baker and I have had a PacBio sequencing project. So thank you, JGI, and thank you, Scott, for being my buddy and longtime companion, colleague. So we work with Cochliobolus, the genus Cochliobolus, which is hypothesized to have arisen a mere 400 million years ago. And I make that point because the fungi themselves are hypothesized to have arisen some 600 million years ago, give or take. So this makes members of the Cochliobolus genus highly similar to each other. And yet, the more than 50 species in the genus vary in their lifestyle. Many are pathogens of cereals, others are non-pathogens. So you can see the collection of species and the various plant hosts that they are specifically pathogenic on. This allows us to do comparative genomics among these species and look, because of the extremely high identity, look for unique regions that we would consider robust candidates for pathogenicity or host specificity. So our model in looking at this group is that there was a progenitor Cochliobolus species that arose approximately 4 million years ago and radiated rapidly to become specialized on various different hosts. We know, using this particular group of fungi, that molecules, secondary metabolite molecules, are key determinants not only of virulence but also of host specificity. For example, T toxin is required for pathogenicity of Cochliobolus heterostrophus to corn. HC toxin, a very different chemistry, is required for C. carbonum to be a pathogen on a different line of corn. We also know that the mechanisms I should step back, that you can quickly see that the chemistry of all of these molecules is diverse, as are the mechanisms. For example, T toxin is a linear polyketide family of linear polyketides produced by a polyketide synthase. HC toxin is a cyclic tetrapeptide produced by a non-ribosomal peptide synthetase. Victorin, its very close relative, is not produced by either of those mechanisms, but rather by RIP mechanism, a ridiculous name, meaning ribosomally synthesized and post-translationally modified mechanisms. So very diverse. The genes which we've identified, as I'll tell you, are unique in each of these genomes, as are the chemicals produced. So the rest of my talk is going to focus on Cochliobolus heterostrophus, a member of the Cochliobolus genus, which in 1970 caused an epidemic of southern cornleaf blight disease. At that time, it was the worst plant disease epidemic on record. The disease appeared in a small area of Florida, and by the end of that summer's growing season had decimated the corn crop on the eastern seaboard, causing great economic loss. Why? Entirely due, as was soon discovered, to the appearance of a new race of the fungus. Now, Cochliobolus heterostrophus, race O, had been described many years before the epidemic, 1925, by Drexler, and it's a very wimpy pathogen of corn, whereas race T, shown on the right here, is devastating to corn. What is the essential difference between race O and race T? It was quickly discovered in 1971, shortly after the epidemic, that race T produces a host-selective toxin called T toxin. It's a family of linear polyketides, and T toxin is specifically active on a particular line of corn. So we had the fungus producing a new chemical, and corn with Texas male sterile cytoplasm, which is male sterile, as the name suggests, and had been discovered in 1940, but by the time of the epidemic was widely planted, a monoculture of susceptible corn. Male sterility trait is encoded by a mitochondrial gene that produces a protein, ERF13, which inserts in the inner mitochondrial membrane and is responsible for the trait of male sterility, which is a terrific trait for farmers and corn breeders, because if you cross-pollinate, you get hybrid vigor in the corn. But as it turned out, that very same protein confers sensitivity to T toxin. So a monoculture, a petri dish, waiting for the appearance of a pathogen. I've already told you this, but so race O of the fungus does nothing to the plant, but race T producing T toxin, the T toxin binds to the ERF13 program and creates a pore in the membrane, and all the guts spill out. So there you had the interaction of a fungus carrying a new set of traits, and the plant with an unusual trait, and susceptibility arose. So our lab and many others have, since its discovery in 1970, have been trying to track down the structure of the locus responsible for T toxin production and the evolutionary history of race T. Where did the DNA encoding T toxin production come from? So I'm going to take you on a chronological romp, a hop, skip, and jump through the decades, as we and others have been using the tools available to us at the time to try to understand the nature of the locus encoding this devastating toxin. So it starts with classical genetics, and it ends today, at least, with PacBio long-read sequencing. And of course, we are very grateful to JGI for assisting us with that. So way back in 1975, Yoder and Grayson used classical genetics to try to understand what Tox1 looked like. And they did it across between a race O and a race T strain. Progeny segregated 1-to-1 for ability to produce toxin and for high virulence on T cytoplasm corn. So the genetic evidence at that point was solidly suggesting that TOX1 is a single genetic locus. Not true, as I'll show you in a minute. Fast forward to the 1990s, and Charlotte Bronson's lab at Iowa State built an RFLP map between a race O and a race T strain, and discovered simple linkage groups, as we would expect, consisting of one race T and one race O contributor, but also noticed a four-armed linkage group consisting of two race O and two race T chromosomes. Race O are shown in red and solid green, and race T chromosomes are hybrid between red and green. That will become clearer in a minute. So this is indicative of a reciprocal translocation between a pair of chromosomes. And to further complicate the understanding or the search for TOX1, TOX1 actually mapped to the intersection of that cruciform structure, which meant that we couldn't tell which race T chromosome TOX1 was on. Was it chromosome 12-6 or 6-12? And just an aside, hybrid chromosomes are named for 12-6, meaning that chromosome 12 from race O contributes the largest part. So fast forward until we had the ability to make mutants in a race T strain and cross them together, and also do electrophoretic separation of chromosomes of highly isogenic race T and race O strains. What we saw was, first of all, that most chromosomes were identical, and that our mutations, our tagged mutations, map to two different chromosomes. So we named those two different chromosomes, or the locus for toxin production, TOX1A and TOX1B. And genetic crosses between mutants also suggested two loci, not one. The other thing that complicated our analysis is that the sum of these race T chromosomes is approximately a megabase more than a simple recombination between 6 and 12 in race O. I failed to say that there's a unique pair in race O, chromosome 6 and 12, and a unique pair in race T. So our tentative model for evolution of race T from race O is that we had a pair of race O chromosomes, 6 and 12. One of them received a large insert of DNA, creating an unstable intermediate that then underwent a reciprocal translocation and distribution of the tox genes on two different chromosomes, 612 and 12-6, inseparably linked from the break point of that reciprocal translocation. Molecular genetic and gene knockout experiments have verified that it requires 10 genes for T-toxin production, 2 polyketide synthases. Not surprising that a polyketide synthase would be involved because this is a linear polyketide. A number of oxidoreductases for decorating the backbone of that molecule, and etc. But the real thing I should point out is that although there are 6 genes at TOX1A, short read sequencing, Illumina sequencing, revealed that pairs of these genes were on very small and unconnectable scaffolds. So we could not connect the 3 pairs of known TOX1 genes using sequencing. TOX1B is a collection of 4 genes captured on a single scaffold sitting side by side. So in addition to its very complex organization, I should tell you that all genes that we've verified are surrounded by highly repeated AT-rich DNA, and they're not found in race O or any other close relative. So, in recent times, Scott Baker and my lab and the JGI partnered to do repeat sequencing of 6 strains, 4 race T strains and 2 race O strains, a collection of highly isogenic lines plus field isolates of wide geographical diversity. So our strategy was the following. Let's imagine that a reciprocal translocation occurred between a pair of race O and a chromosomes, creating a unique pair in race T. So originally we'd have RFLP markers shown in purple, 395 and B88, linked on the same chromosome to each other, but also flanking the theoretical breakpoint, and ditto for chromosome 6. If those chromosomes undergo a reciprocal translocation, then new linkages are formed, and 395 is now no longer linked to B88, but is linked to B149. So if we can locate scaffolds that carry both of these markers, 395 and 149, we will have captured the gene space between them and the tox genes, as well as the reciprocal translocation breakpoint. So, wildly successful. Chromosome 12.6 was located on a gigantic scaffold of more than 2.6 megabases. That scaffold carries the flanking RFLP markers that we predicted would be there, and it also encompasses the 6 known TOX1 genes at TOX1A. For the first time, we have the order of those genes arranged thusly. But the story gets even more complex because the gene space taken up by these 3 pairs of tox genes is separated by hundreds of kb. So the tox genes are actually pairs, little islands in a sea of non-coding DNA. TOX1B was similarly captured on a large chromosome, and we didn't learn anything more about the organization of the locus because we already knew that the genes were side by side, flanked by these markers. So physical organization of the TOX1 locus, I'll go back to the 4-armed linkage group consisting of chromosome 12 and 6 from race O. Here's a blow up of a cartoon of what we've found by long read sequencing. Well, the first thing we found is that there are 3 giant loops of DNA that is race specific, that are race specific, carrying islands, little islands of the tox genes in race T. The corresponding positions in race O are not single nucleotide changes, but rather 1.6 kb of race O specific sequence on chromosome 6 and 315 base pairs of sequence on chromosome 12. In addition, it gets increasingly complex as there's 5 on chromosome 12, there's 5.5 kb of conserved DNA between race O and race T, and then 2.3 kb of race O specific DNA corresponding to the loop out in race T. So a different, easier way to see this, if we compared hybrid chromosomes 12-6 and 6-12 in race T to those in race O, is that we have 3 giant loops of enormous amount of race T DNA with little, as I said, islands of tox encoding DNA. So I see I have a minute or two. I thought I would tell you about our attempts to track the evolutionary history of the tox genes, which I've already told you are extremely complex, not side by side as most biosynthetic gene clusters have been found to be. So thanks to the wealth of genome sequencing, we've been able to identify tox genes in several unrelated species. And in all cases, those genes are in compact gene clusters. In addition, smack in the middle of those compact gene clusters is an ABC transporter gene and a different oxidoreductase gene. Those ABC and OXR genes are not found anywhere in Cochliobolus. So we think at this point that a compact collinear cluster is the likely ancestral condition, because it's very hard to imagine that the disorganized cluster or set of genes in Cochliobolus gave rise to an organized cluster in disparate organisms. We also think that the extra genes in the clusters suggest a different metabolite may be made in these other fungi. And we can also say with assurance that the TOX1 cluster was not acquired by race T via horizontal transfer at the time of the epidemic from any species sequenced so far.
Awesome. I think we probably should leave it there because we're